Good afternoon, everyone. I am your host Alok Kumarja, and I warmly welcome you to today's webinar. We will be starting shortly, so thank you for your patience. Good afternoon, everyone. I warmly welcome you to today's webinar. We would be starting shortly, so thank you for your patience. Good afternoon, everyone. Once again, I hope you all are doing well. On behalf of Advent Informatics, it's my pleasure to welcome you to today's exciting webinar. Our topic is both fascinating and highly relevant: mastering the 3D ligand editor of Molsoft, advanced tools for ligand design and optimization. We are thrilled to have such an enthusiastic audience joining us today. We look forward to insightful and engaging session ahead. Before we dive into today's topic, let me take a moment and introduce you to Advent Informatics. Advent Informatics Private Limited is a Pune-based company specializing in distribution of life science softwares. We provide cutting-edge software solutions in the fields of chemoinformatics, molecular modeling, medicinal chemistry, chemical data curation, chemical and biological data management, etc. Our clients include many of the leading pharmaceutical, chemical, and CRO companies in India, as well as academic institution and government research organization. Our mission is to empower Indian researchers and students by providing access to world's renowned software tools. Currently, we represent solutions from industry leaders such as Molsoft LLC, Chemexon KFT, Chempass, Certara, Transgen IT Solution, and Keymodel. Now, let's dive into today's exciting topic: mastering the 3D ligand of Molsoft, advanced tools for ligand design and optimization. Molsoft is a global leader in molecular modeling and drug discovery, known for its innovative solutions that empower researchers worldwide. In this webinar, we will explore the 3D Ligand Editor, a state-of-art tool designed to revolutionize the way researchers design and optimize ligands. You will discover advantage, how its advanced features can help streamlining your workflows, enhancing the precision and accelerate the drug discovery process. Whether you are designing new ligands or optimizing existing ones, this tool offers unparalleled capabilities to evaluate your research. Now, it's my pleasure to introduce you to today's speaker, Dr. Pathan Mohsin Khan. Dr. Khan has been an application scientist at Advent Informatics Private Limited for past 1.5 years now, where he has been instrumental in supporting our clients and his expertise. Prior to joining Advent Informatics, he worked as a senior R&D specialist at QSAR Labs and in turn as a research data scientist at Edelweiss Connect in Basel, Switzerland. Dr. Khan holds both a doctorate and a postgraduate degree from National Institute of Pharmaceutical Education and Research, NIPER, Kolkata, showcasing his strong academic foundation. With 12 research articles published in peer-reviewed journals and 3 book chapters, with renowned publishing houses, Dr. Khan has made significant contribution to this field. His expertise spans structure-based, ligand-based drug design techniques, including protein modeling, binding site predictions, molecular docking, pharmacophore modeling, QSAR modeling, and virtual screening. If you have any question for Dr. Khan during his talk, please feel free to submit them in Q&A section. You can find the Q&A option in the toolbar at the bottom of your screen. Now, without any further delay, I hand over the floor to Dr. Khan. Dr. Khan, the stage is yours.
So thank you, Alok, and thank you everyone for joining us for today's webinar on mastering 3D ligand editor of Molsoft, which is an advanced tool for ligand design and optimization within the binding site of protein of interest. So in this webinar, we will discuss about ICM 3D ligand editor, or you can say interactive ICM 3D ligand editor. So this editor is widely used throughout the pharmaceutical and biotech industry for ligand design and optimization within the binding site of protein of interest. This tool was built in collaboration with the medicinal chemist at Novartis. And it is fully interactive tool where one can make changes either in 2D or in 3D of their small molecule and immediately see the effect of those modification on the binding score of that particular ligand molecule towards the receptor protein. So we will use for demonstration for demonstration of this ICM 3D ligand editor, we will we will use particular protein of interest and see how we can perform different types of modification and see the effect of those modification on your ligand molecule immediately by using ICM 3D ligand editor. And this particular ICM 3D ligand editor is already published in Journal of Chemical Information and Modeling. Now, if you'll talk about the application of this ICM 3D ligand editor, there are a number of applications in which the ICM 3D ligand editor has been widely used. For the first one is finding base replacement group for your ligand molecule. Let's say if you have a standard ligand molecule and you want to replace some functional groups with some other functional group and see the effect of those functional group on the binding interaction of your ligand molecule within the binding site of protein of interest. So we have a set of functional groups available. Like if you want to replace some lipophilic group with some other small lipophilic groups, then we have a data set already available data set in Molsoft to replace with. Then if you want to replace some donor group with some other donor groups from the database, it will also replace them. Then we have also small acceptor, positive charge, negative charge, small hetero ring, even a small fused hetero ring, which you can replace with your functional groups, whatever you want to replace from the standard molecule. So these are the different functional groups. As you can see, a small lipophilic, small donor, small acceptor, positive charge, negative charge, hetero ring, fused ring. And after replacing this particular functional group, it will calculate two binding score. One is RTCNN score, which is a neural network score provided by Molsoft for predicting the interaction between protein and the ligand molecule, and then ligand efficiency score for your generated ligand molecule. Then similarly, the other application for this particular tool is to perform multi-position R group scanning. Let's say you want to modify multiple position on your ligand molecule. If you have a standard ligand molecule similar to this way, and you want to add certain functional group on multiple position of your ligand molecule, then you can use this multi-position R group scan, and it will add those multiple groups on the multiple position and generate novel ligand molecule, and subsequently calculate the binding score of those generated new molecule. Similarly, you can also perform hetero scan, which is like if you want to replace some carbon group with nitrogen, oxygen, or sulfur in your standard ligand molecule and see the effect of those changes in the ligand molecule. You can also perform on those type of study by using ICM 3D ligand editor. Similarly, the other application of ICM 3D ligand editor is to perform bioisostere scanning. So in drug design, as we all know, bioisostere can be used to address toxicity, change bioavailability, alter metabolism, and change activity of any lead compounds. So basically, the bioisosteres are chemical substituent with similar physicochemical properties, which produce broadly similar biological properties to another chemical compound. So if you will see here in glutamic acid, this is the functional group. If you will look for this bioisostere scanning by using ICM 3D ligand editor, it will provide you a similar bioisostere for your ligand molecule in the form like ibotenic acid. And these are the different studies which has been performed by using ICM 3D ligand editor bioisostere scanning. This particular functional group has been replaced with this particular functional group, and the Ki value has been increased. Then you can also use ICM 3D ligand editor for core replacement. Let's say if you have a standard ligand molecule and you want to replace the core of your ligand molecule, then it will either use ICM 3 atomic property field or ICM pharmacophore model to identify similar core in the available 2D or 3D database. It lets you to interactively select two or more point of connection. Like if you have this one is the point of connection, and then another one, this one is the point of connection. If you have more than two connection also, you can select those connection and search in the either 2D database or in the 3D database. And this similarity search for for your scaffold will be based on like either atomic property field or either based on the pharmacophoric points. So basically, ICM 3D Ligand Editor is very helpful for single point modification, multiple point modification, heterogroup scanning, bioisostere scanning, as well as scaffold hopping or core replacement. Now, to perform modification on your ligand molecule in ICM 3D Ligand Editor, you have to have a knowledge about some ligand receptor representation. The first one is receptor representation. So in the receptor representation, the protein of your the binding site of your protein of interest is represented based on the binding properties of the residues available in the binding site. So let's say if you have a green color surface on your in your protein binding site, so there is a chance there is a that color represent that hydrophobic residues present in the binding site, and red color represent the hydrogen bond acceptor, and blue color represent hydrogen bond donor. Similarly, ligand surface. So this bindings receptor surface is very helpful for identifying the open cavity and closed cavity in the binding site of your protein of interest. Similarly, you can also have to perform like you have to generate a ligand surface, which is also very helpful to which will also allow you to identify where is the space available in your binding site to grow your ligand molecule. Then similarly, you can also optimize your ligand molecule, standard ligand molecule within the binding site. Then you can also generate a pretty view, which will allow you like which residues are interacting with your protein of interest in the binding site with the ligand molecule. Similarly, you can also calculate the atomic energy circle or atomic energy circle for each of the atom of your ligand molecule, and see whether that particular atom of the ligand molecule is having or forming any clashes with the receptor protein and or not. Then similarly, you can also identify which are the unsatisfied hydrogen bond atoms in your ligand molecules, which can be replaced or which can be involved in hydrogen bond interaction and increase the binding score of your ligand molecule within the binding site of protein of interest. So these are the different visualization which we will see in the demonstration as well. Then we will use for this particular demonstration, we will use CDK5 protein, which is the PDB-ID for that particular protein structure is 1-UNL, which is a complex with the roscovitin. So it is an experimental drug candidate in the family of cyclin-dependent kinase, roscovitin, and preferentially inhibit multiple enzyme target like CDK2, CDK5. CDK5 is the default, CDK2, CDK7, and CDK9, and it will alter the growth phase or state within the cell cycle of treated cell. So we will see how we can perform modification on this roscovitin ligand molecule within the binding site of protein of interest by using ICM 3D Ligand Editor, and generate novel ligand molecule like by using single modification, or by using bioisostere scanning, or by using core replacement. We will see how we can generate different novel ligand molecule from the standard ligand molecule. So let's move to demonstration. If you have any question from the presentation, you can ask me.
Sir, we have received multiple questions till now, but we would be taking them after your session is completed together.
So first of all, if you want to perform ligand modification, or if you want to perform ligand design optimization by using ICM 3D Ligand Editor, so for that you need your ligand molecule and the protein should be in the complex form. That complex either can be a experimental structure, or it can be a complex which is obtained by docking study. So the complex which we are using here is 1-UNL, which is a experimental crystal structure of protein complex with the ligand molecule. So you just have to, in Molsoft, you have direct access to multiple databases through this search tab. If you will see here, 3DB search. NCBI Blast, Uniprot database. So all these databases you can directly access from the graphical user interface of Molsoft. So you just have to select the PDB data bank and provide the PDB ID for the particular protein structure which you want to import. So I'll just type one UNL, which is a PDB ID, and press enter, and it will automatically fetch that particular protein structure from the PDB data bank. Now, if you will see here, we have imported the this one UNL object from the PDB data bank into ICM graphical user interface. And you can see this particular structure is based on X-ray crystallography, comprises of, and the resolution of this particular structure is 2.2 angstrom, and it consists of multiple chain A, if you will see here, then roscovitin, which is our ligand molecule, which is bound in the chain A, and then other subunits of this particular crystal structure. So if you want to remove the other subunits, you just have to double click and select them, and just press shift and hold to select multiple entries, then right click and you will see this option here, delete, and you just have to press yes, and it will automatically remove that particular ligand molecule, particular entries from the same object. Now we have only the protein structure and the ligand molecule and the water molecule associated with the protein of interest. So after importing after importing and removing the unnecessary chain, you just have to go to this Lig Edit button over here, which will allows you to access for ICM 3D Ligand Editor. So in the Lig Edit, you will have multiple buttons. The first one, if you will see here, Setup Ligand. So you just have to select this Setup Ligand option, and it will automatically select ligand molecule from your object file, whatever object file or whatever complex file you will have in the workspace panel. So here you will see within this one UNL object, we have this ARRCC, which is a roscovitin, which is our ligand molecule. If you will see here, it will automatically select that particular ligand molecule. If you will see here, current setup, ligand molecule A_1 UNL dot ARCC, which is A underscore basically represent object. Then the name of that particular object is one UNL. And then within that particular object, there is an entry which is ARRCC, which is representing the ligand molecule. Then you can also auto assign formal charges. Let's say if you want to charge this particular ligand molecule at particular pH, you can specify that pH over here. By default, it will be charge your ligand molecule with a 7 pH, and you just have to press OK. And it will start converting your ligand molecule into ICM object and create a separate object for your ligand molecule within the workspace panel. So you'll just have to press OK, and you can see it has created the separate object for your ligand molecule. Now if you'll see object in bracket 2 atom 1 UNL underscore lig, which is our ligand molecule. If you'll see here, if you'll see, this is our ligand molecule. It has created separate object for the ligand molecule. Now, if you will see the next option in the Lig Edit, the next button is the Setup Receptor option. So you just have to open click on this button, and it will open up this Setup Receptor window, where you will see the current setup A_1 UNL underscore lig, which is for our ligand molecule, 1 UNL underscore lig, which is generated ligand molecule. Setup receptor setup has not yet set up. So you have to select the receptor protein. So here if you see define receptor object A_1 UNL. So if you'll see here A_1 UNL, A_1 UNL, which is our protein structure. Then it will ask you whether you want to remove the water molecules which are present in this particular structure, or whether you want to keep those water molecules. So you just have to select options related to options based on your requirement. So I'll just delete all the water molecules. Similarly, this is an X-ray crystallographic structure. So I will add hydrogen atom, optimize hydrogen atom, optimize different residues. If any side chain is missing, I will add those missing side chain, or I will automatically build those missing side chain by unmarking this particular option. And then you just have to press on this button here, make box around existing ligand molecule, and you will see it will create a grid box around your co-crystallized ligand molecule at in the binding site of protein of interest. And if you want to increase the size of this generated grid box, you can increase the margin, like, and this particular box is around 3 angstrom. If you want to increase it to 5 or any other, you just have to type it over here, and then you just have to press OK, and it will start generating a grid map for our protein of interest binding site grid map of grid map for our protein of interest. And after generating that particular grid map, it will add our generated grid map within the workspace panel with the same, with the different object with the name you will see here, 1UNL underscore race 1, which has protein structure, and the ligand molecule is represented with the ligand surface. So if you will see, this is our protein structure, prepared protein structure, and this is our original structure which we have used to generate this separate separate receptor object and separate ligand object. Now if you will see, this is the setup. This is for like if you will see, this is the ligand surface on the surface of your ligand molecule. But in this visualization, we are not able to see which residues are forming any type of interaction and how we can grow that particular ligand molecule within the binding site of protein of interest. So for that, first of all, you just have to go to again your Ligand button over here, Ligand tab. In the Ligand tab, there are multiple options available. So here you will see this option, toggle various ligand properties. So you just have to click on this drop down menu and it will open up multiple options. So here first you have to select pretty view, which will show you the pretty visualization for your protein ligand complex. You can remove the CPK representation from the ligand molecule. You just have to click on this toggle in the display tab. You just have to uncheck this button, toggle CPK representation, and it will automatically remove the CPK representation. Similarly, you can make your protein structure transparent so that you can perform visual analysis clearly for modification on your ligand molecule. Now, again, you just have to go to Ligand Edit button. Then within the same toggle on and toggle various ligand properties, you can visualize like receptor pocket, which will give us which binding with which will give the receptor pocket based on the binding properties of each residues, and it will also give us idea whether the binding pocket is closed or open for any modification. So if you will see from this visualization, we have a two position available to grow our ligand molecule. One is at this particular phenyl ring and another one is at this particular ethyl functional group. But if you will see, if you will add any functional group at this particular, let's say here nitrogen, or at this ethyl group, there is no available space in the binding site to grow any to add any functional group at this particular position on the surface of your on your ligand molecule. The next option, the next visual analysis you can perform by using our ligand editor ligand surface. So you just have to visualize your ligand surface, and ligand surface also allows you to modify your ligand molecule or visualize the open cavities available for binding site, open cavity available for modification. So if you will see at this ethyl group, there is no open cavity available, but if you will see at this benzene ring, there is a open cavity available for addition of functional group. Similarly, at this position also, if you will see, there is an open position to grow our ligand molecule within the binding site and improve or add certain functional group which can improve the active binding score of our ligand molecule. So this is the second way you can perform binding analysis, binding site analysis of your protein of interest to design novel ligand molecule. Then next, you can also visualize unsatisfied hydrogen bond atoms on your ligand molecule. So you just have to click on this button here, and it will show you the atoms which are not forming any type of hydrogen bond interaction with the residues in the protein of interest. So either you can like replace those atom with some other functional group, or you can remove those particular functional groups with some other functional groups which can involve in different types of interactions. Similarly, you can also relax your ligand molecule within the binding site of protein of interest. So you just have to click on this button here, display relaxed ligand molecule, and it will show you the relaxed ligand molecule in comparison with your co-crystallized ligand molecule within the binding site of protein of interest. Similarly, you can use this button here, display atomic energy circle. So it will provide you the energy for each atom on your ligand molecule. If this energy of your ligand molecule atom energy will be in red, that particular atom will be forming any clashes with the protein of interest. So we'll see how we can visualize that clashes also by adding some functional group on the position where there is no space to grow our ligand molecule, and how they can form, they can involve in like clashes between protein and the ligand molecule. So this way we will perform visual analysis of our binding site. Now we have identified multiple position on the surface of our protein structure. So we'll just visualize all the hydrogen atom on the ligand molecule as well as the protein structure, so that we can add set of functional group to perform to see the visual to perform our modification. Let's say we have seen we have from receptor pocket as well as ligand surface, we have found out that at this particular position, addition of any functional group, there is no space to grow our ligand molecule. But let's say if we will add any functional group at this particular position, what will what will be what that particular functional group will cause. So if you'll see the here you have a Artesian in score, which is a calculated score for this particular complex within the binding site, which is -37 by default, -37. Now we will use the editor panel to add certain functional group at this particular position. So you will see the third option here is the edit tool panel. So you just have to click on this edit tool panel and it will open up this particular window. So here you will see the first one is like your periodic table. If you want to replace some atom with some other atoms, so you can use this periodic table. If you want to add some functional groups, so we have these options available, default options, some functional group, default functional group. We are also having a database of around 60,000 functional groups available in the advanced group scan, so that like you can add those functional group at particular position, at single position, and generate 60,000 novel molecule with a different functional group within that particular position of interest. So now let's say if you want to remove particular functional group, let's say we want to remove, we have seen this particular hydroxy group is not involved in any hydrogen bond interaction. What if we will remove this particular functional group? Whether that particular function removal of that particular hydroxy group will increase the binding score, or whether it is reduce the binding score. So we'll just use this eraser button from here, and then select on that particular atom or particular functional group which you want to remove, and it will automatically remove that particular functional group. Now if you will see, we do not have a hydroxy group in this particular position, and we will see now the score is not calculated for this particular ligand molecule. This is a modified ligand molecule. So to calculate the score, you just have to click on this recalculate binding score, and you will see the previously the binding score is -37. Now it is improved to -38. So removing that particular functional group from the protein of interest, from the set of from our standard ligand molecule, is increasing our binding score for our ligand molecule within the binding site. Let's say you want to go back to your original ligand molecule which has this particular hydroxy group. So you can use this undo and redo button here. So you just click on this undo erase atom, and it will automatically get back to you to your hydroxy group. And if you will see, with hydroxy group, the binding score of this particular ligand molecule is -37.34. So this is what we have removed the particular atom for functional group from the standard ligand molecule based on like unsatisfied hydrogen bond interactions, or not involving any type of hydrogen bond interaction within the binding site of protein of interest. Now we have seen like if we will add any functional group at this particular position in the ligand molecule, it will cause it, there is no space to grow our ligand molecule at this in this direction. So let's say if we will add, what will happen? So let's select this particular functional group or any other functional group. Let's say I will select this particular functional group. So you just have to select that particular functional groups and move your cursor and select the particular hydrogen atom on which you want to add those add that particular functional group. So I'll just add that particular functional group over here, and it will start generating different conformation of that particular after adding that particular functional group within the binding site of protein of interest. So now if you'll see, this particular functional groups is causing clashes, which is represented with this brown zigzag color. And if you will calculate the binding score of this particular ligand molecule with this particular functional group, with this button here, recalculate binding score, you will see there is huge reduce in the binding interactions. If you'll see binding score from minus 37, it became minus 2. So this particular addition of any functional group at this particular position will leads to decrease in the binding interaction of your ligand molecule. So you are not now you are now we are satisfied that at this particular position we are not able to form any type of interaction. I add any functional group on the ligand molecule. So if you want to go back to your original ligand molecule, you can undo this. Just click on this button here, and it will go back to your original ligand molecule, and you will see the RTCN score or the binding score for your ligand molecule is minus 37. Let's say if you want to add from now, let's say we have performed visual analysis receptor pocket, and we have seen that this particular position on the ligand molecule is available for growing our ligand molecule. So let's say we will select multiple ligand molecule, multiple function group in a single way and generate set of ligand molecule. So I'll just select these functional groups. You can select, or you can select here also some sulfonamide. And then you just have to click on that particular functional group, particular hydrogen atom on which you want to add those functional group. So if you want to add at this particular position, you just have to click on that particular position. Or let's say first single molecule, if you want to generate, let's say you want to add this sulfonamide at this particular position and see the effect of that particular functional group on binding score. So you just click on that and it will identify that particular functional group. Now we have generated novel ligand molecule with this particular functional group at this particular position. You can calculate the binding score. It is reducing. So this sulfonamide, addition of the sulfonamide is reducing the binding score of this particular ligand molecule, although there is a supposed to grow this particular ligand molecule at this particular position. So you can go back to original. You can add some other functional group, let's say cyclic ring on this benzene. Let's say we'll add this particular functional group benzene and calculate some again calculate the binding score of this particular generated novel molecule. It is also reducing the binding score. But let's say now if we will replace some functional group, some carbon atom with the nitrogen atom within this particular added hydrogen added ring, so then what will be the changes in the binding interaction? So let's say we will change this carbon with the nitrogen atom. You want to change this carbon atom with the nitrogen. So just have to click on this button here, and it will add that nitrogen atom within the binding within the ring. And if you will calculate the binding score, you will see the binding score has been slightly changed. Similar way, you can add some other functional group and generate multiple ligand molecules, or you can go back to your original ligand molecule. Like you just have to click on and go back to your original ligand molecule. And if you want to generate a series of molecule from this multiple functional groups, so you just have to select multiple functional groups here, and let's say some sulfonamide, and click on that particular hydrogen atom, and it will start generating novel ligand molecule with multiple function group which we have selected to generate novel ligand molecule. And after generating those novel ligand molecule, you will have a table which have your score for your original ligand molecule as well as score for each of the generated or modified atom at that particular position in your ligand molecule. And you can see whether that addition of single ligand addition of that particular functional group is improving the activity, improving the binding score, or reducing the binding score. So here you will see this is the green region represent our original or standard ligand molecule, and this is the region which we have where we have added this hydroxy group instead of hydrogen atom, and the binding score is minus 37. Similarly, if you will see our original ligand molecule, which is with hydrogen atom. Here you will see the docking score is -30, -27, and artisanal score is -30. And then if you will have binding score greater than that -30, you will consider that modification will be feasible. You'll see here we have this particular 32, which is feasible. If you will add, if you want to visualize, you can add that particular functional particular ligand molecule and visualize within the binding site of protein of interest. Similarly, you can also visualize other like this 31, which is also having better score than your standard ligand molecule. Similarly, you can also see this particular functional group is also having multiple better score than your standard ligand molecule. So this way you can visualize multiple ligand molecules or modify and generate novel ligand molecule by using single point modification in your ligand molecule. If you want to add this generated novel ligand molecule into new chemical table so that you can further perform docking analysis and all those things, so you just have to click on this button here, add molecule, add current ligand to table. So you just have to click on that and it will add that particular ligand molecule into current table. Let's say if you want to visualize this particular pose and click on this, and then you will go to ligand, you will see this is the second ligand molecule which has been added to our table. So this is a one way you can perform perform modification or add certain functional group on your ligand molecule within the binding site of protein of interest. Similarly, let's say if you want to add your own functional group, then there is an option in the editor panel, add your own functional groups and design novel ligand molecule by adding those functional group on your ligand molecules. So this way you can perform single point modification. Let's say if you want to replace particular functional group, go back to our original ligand molecule. So this is our original ligand molecule. Now let's say we want to add some functional group instead of particular functional group and scan like database already available with us. So for that you just have to go to this option here, scan, and there is option here, find groups or find linkers. So you just have to select this find groups and it will open up this option here, database scan. So if you'll select particular item on your ligand molecule, let's say you want to replace this particular item or add any functional group at this particular item, or let's say you want to replace this just a minute. Let's say you want to add particular functional group, so you just have to use this atom selection button, select that particular functional group. Now at this particular position, if you want to add a small lipophilic group, small donor group, small acceptor, positive charge, negative charge, small hetero ring, or small fuse ring, so all these databases are small prepared databases of functional groups are available in the Molsoft. So with those functional groups you can generate new ligand molecules. So by combining all these functional groups, we have around 60,000 functional groups. Let's say you want to add only small lipophilic, so it will generate a data set, it will generate novel ligand molecule around 283. Let's say you want to generate add some small donor group at particular position, then you just have to click on this button here, small donor, and it will generate around 203 2,300 novel ligand molecule on your binding for your ligand molecule. Similar for acceptor, there is a 3,000. Then for positive charge, there is 18,000 negative charge. So for a small fuse ring like for 4,400, you can generate novel ligand molecule with fused ring. Let's say if you want to replace this particular ring, which is like here we have this benzene ring. If you want to replace this benzene ring, so you just have to first click on this button here, and then you can select this button here, click on this particular bond which is linking this particular ring with the entire protein entire ligand molecule. So you just have to select that particular structure pointing towards the ring. Now you can go to advanced scan and small, you can select this small hetero ring, let's say, and it will replace that particular selected ring with those multiple structures, and you just have to press OK, and it will start generating like 7,600 novel ligand molecule with different small hetero rings. Similarly, let's say if you want to perform bioisostere scanning, just go back to original. Let's say you want to perform bioisostere scanning. So if you just have to select that particular functional group and look for similar bioisostere. Let's say you want to replace this particular methoxy group, this particular group with some other functional group. So you just have to go to this button here. First, you have to select that particular functional group. Click on this button. Select that particular functional groups pointing towards the original ligand molecule. So we have selected this functional group and we want to search for similar bioisostere. So you just have to go to this advanced option here. Click on this bioisostere scan. Either you can search for similar bioisostere in the ChemBL database of ChemBL or from PDB databank. So you just select that particular database and how many hits you want. You can specify that particular number over here and just press OK, and it will start looking for similar bioisostere to your this particular selected functional groups and provide you novel ligand molecule with the different with the similar bioisosteres in terms of physicochemical properties. However, the chemical structure will be different. So you can see it is trying to generate novel ligand molecule with a different bioisostere for our ligand molecule. And after completion of this particular job, it will give you the results modifier table, which will have different novel generated molecules with different bioisosteres. And you can see and compare the generated binding score with your original ligand molecule. If the score is improving, then you can select that particular modification for further ligand optimization. Yes. Can select the particular function. Similarly, if you want to replace particular groups or particular scaffold with some new scaffold, if you want to replace some scaffold with new scaffold, so you just have to select that particular scaffold by using this scissor button over here. Click on this button, or like the bonds which are connecting that particular scaffold with the rest of the chemical structure. So you just have to select that particular bond first. Then second, here if you'll see this particular scaffold, and then third one, this one. Now we want to replace this particular scaffold with some other scaffold. So for that, you just have to go to advanced, and you will see here scan fragment, or you can search for linkers, find linkers, and click on, like, you can download that linker database from the Molsoft website and generate novel ligand molecule. Similarly, you can also identify groups which can be replacement for this particular functional groups. So this way you can select that particular scaffold and generate novel ligand molecule in your in your database on your ligand molecule. Now let's say if you want to replace any functional group on this functional group, let's say you want to replace this particular functional group with similar biosystem. So you just have to select that and click on any database and click on that, and it will start generating novel ligand molecule with different biosystem. And after completion of the job, you will see which ligand molecule is improving the binding score, and which bioisoster is improving the binding score, and which is reducing the binding score. And there are multiple other options are also available in Ligand Editor, ISM3D Ligand Editor. Like after generating novel ligand molecule, you can dock or redock those ligand molecule within the binding site of protein of interest by using this option available here, like dock immediately option available here. In advance also, there are multiple options, like if you have a covalent ligand molecule, you can also generate novel ligand molecule with the covalent docking. You can also dock by using covalent docking protocol. Then you can also generate 2D interaction diagram for your protein ligand complex. You can add them to new chemical table. So So if you will see, this is the modifier table with the different scaffolds. So we have replaced that particular ethyl group with some other functional groups. If you will see, the standard our its ligand molecule is, which is having entirely green. So there are multiple functional groups which we have added and calculated the RTCN score. We'll see. If you want to visualize this particular binding within the binding site, you can visualize them and see which one is having better score than your ligand standard ligand molecule. So if you'll see, instead of this particular ethyl group here, like here we have this cyclopentyl ring, then this methyl group, then trifluoromethyl. All these functional groups are replaced as a replacement for these ethyl groups. Bioisostere are replacement for your ethyl groups. And if you want to add this ligand molecule, so you just have to click on this button here, ligand add to current table, and it will automatically add it to your current table. And after adding modified, like based on the RTCN score, you have found out that particular residues are having better score. So you have added them to novel ligand molecule. Then you can go to your advanced option here and dock that ligand table, whatever you have added ligand molecule. So you can dock all of them within the binding site of protein of interest, and also perform visual analysis of your docking results, like considering different other scoring function like ICM score and MF score, and different interaction energy terms like hydrogen bond interaction energy, van der Waal interaction energy, dissolution energy, solution energy, etc. Similarly, you can also generate 2D interaction diagram from this button here, like generate 2D diagram for your protein ligand complex. So I believe this is it, by which this is it from the today's webinar, like more or less from today's webinar, which will be on which was on ICM 3D ligand editor, which is more useful for ligand design and optimization within the binding site of protein of interest. I have not shown you how you can perform changes by using. We have seen only the changes by using 3D. So if you want to perform changes in the 2D and see the effect of those changes on 3D, so I'll just go back to original. Yeah. So if you will see in this editor panel, there is an option where you can go use that particular option to perform changes in the 2D. So if you will see, this is the option, editor, edit or create new ligand in 2D molecular editor. So just have to open, and it will open up your ligand molecule within the 2D chemical editor. Let's say you want to add this particular functional group at this particular position, methoxy group, or you want to add that chlorine at that particular position. You just have to click, after modification, you just have to click on that apply changes to 3D, and it will automatically add that changes to 3D. If you will see this particular chlorine which we have added at this particular position. Similarly, you can calculate the binding score of that particular pose and all those things similar to any other which we have performed in 3D. So this is more or less from the ICM 3D ligand editor, which is more useful for ligand design and optimization within the binding site of protein of interest. Is there any question?
Yes, sir. Thank you so much, Dr. Khan, for your wonderful presentation and demonstration. Now we will move to Q&A session. If you have any question for Dr. Khan regarding today's topic, feel free to ask them. To start off, we have already received a few interesting questions from participants. So the first question is, how can I incorporate the modified ligand into existing table for subsequent docking studies?
So as I have already shown you, and just have to select that particular row, and you will see this option here. add current ligand to table, and it will automatically add that particular ligand molecule, let's say this ligand molecule, to your current table. You will see at the end that particular ligand molecule has been added, and then you can dock directly this particular table by using your generated grid file. Is there any other question?
Yeah. Can polymorphism of ligands be detected?
Polymorphism in what sense, like while designing?
Unfortunately, they have not described it any further detail, but yeah, this is the question.
So while designing, it won't be detected, I think. But when you are performing docking and all this thing, at the time you can specify that.
Okay. And how does ligand surface assist in ligand modification using the ICM 3D ligand editor?
Ligand surface.
Yes, I'll
So
repeat the question. How does the ligand surface assist in ligand modification using the ICM 3D ligand editor?
so basically, ligand surface allows you to visualize available cavities present on your ligand molecule to grow your ligand molecule.
Okay. What does atomic circle energy represent in ICM ligand editor?
So basically, atomic circle energy is the individual atom energy of your ligand molecule, and overall contribution of that atom in total energy calculation of that particular ligand molecule. If that is in like and that's shown in green, which means that particular ligand molecule is not causing any type of clashes with the protein of interest. If that's shown in red, so you can see there will be a that particular atom or that particular function group will form will cause a clashes with the protein of interest.
Thank you all for the wonderful questions that came in during the session. We truly appreciate your active participation and engagement. Unfortunately, due to time constraints, we aren't able to address all the questions live. But rest assured, every unanswered question would be reviewed, and our team will get back to you via mail with the detailed responses. With that, we have reached the end of today's webinar. On behalf of Advent Informatics, I'd like to extend my heartfelt thanks to Dr. Patan Mochin Khan for sharing his expertise and delivering such an insightful and engaging session. A big thank you to our audience as well as for your time, attention, and thoughtful questions. Your participation made this event a success, and we look forward to hosting you in our upcoming events and webinars soon. Thank you.